Say you are doing some lead optimization. When will you go for isoster approach, and when for bioisoster approach?
When we want the same biological activity, we can go for bioisosters.
So when we prefer for isoster?
For better interaction along,
When we want to refine the activity, sir.
Activity in the sense, which activity?
you mean functionality will be different in isoster?
Yeah, I completely understand your answer regarding bioisoster. So my question is, when what's the need for going for isoster?
For optimizing the lead compounds.
Those are very basic things, right? So, what is the situation in which we prefer to go for isoster replacement?
Leads to controlled changes in like steric electronic properties.
Absolutely right. Okay, so who answered this?
Sir, Sai Ganapriyas.
Okay, that's correct, right? So if you want to make a very controlled, electronic replacement, then we go for isoster. If your intention is, preserving the biological activity, then we should go for bioisoster. So here is example. So I think this I already discussed, right? So pyrrole ring is, a pyrrole ring and amide group. So this is amide. This group is amide group, CONH, and this pyrrole ring. So replacing the amide by pyrrole or pyrrole by amide, we can retain the biological activity. So we can say that amide functional group and pyrrole ring, we can call them as bioisoster. So next is the simplification. Uh, so Okay, so what is simplification? Actually, what's the need for simplification? Simplification is very commonly used approach in lead optimization. Is my voice clear? Any
Yes,
feedback
sir.
noise? No feedback noise, right?
Yes.
Okay, fine. So why we go for simplification? The main reason is as we already discussed that natural compounds are one of the major source of drug-like molecule. So we discussed about three important sources. One is the natural based molecule, another one is synthetic, and the another one is virtual world. Am I right? Okay, so if you take any natural compound, right? So basically, if you look at the structure of natural compounds, uh, you know, you can find some complexity in the structure of almost all the natural compounds. What I mean here is the structure of the natural compounds are not so simple. That is the way how the nature it makes the compound, right? So if you take the structure of morphine, or if you take we have so many terpenoids, right? So if you look at the structure of those small drug like molecules, definitely the structure won't be very simple. We can say we can see some kind of complexity in the structure, right? The problem with complex structure is, in small scale, we can extract those molecules from plant sources from the nature. But if you want to go for large scale, say your molecule is approved by FDA to market it as a drug molecule, we cannot completely rely or depend on nature. Okay, because the amount of drug or drug-like molecule that we can isolate from nature, the amount will be very less. So we cannot completely rely on nature. So obviously, what we have to do is, even though it's a natural compound, so we have to synthesize the same molecule in laboratory condition for large scale, and that is called as large scale synthesis. So as I said, because of the complexity of the natural compound, we cannot easily synthesize those compounds in laboratory. It's a very difficult process, right? So in such cases, we go for a kind of optimization called as simplification, right? So here, say for example, assume that this is a natural compound. Okay, you are interested on. So if we ask a question whether this compound is a simple compound or it's a very complex, we can say it's a very complex structure because it's having so many number of functional groups. You can see OH group is there, NHMe is there, OMe is there, Cl, pH, COOH, right? So almost 6 functional groups are present in one single compound, right? So definitely it is not going to be a very simple compound. Definitely synthesizing this compound is not going to be very simple on laboratory condition. So what we have to do is we have to simplify the structure and take that structure for synthesis. That process is what is called as simplification process. So this is the initial lead molecule, and we can say this is after simplification. So after simplification, how many functional groups we have in the drug molecule? We have only 3 functional groups. Am I right?
Yes, sir.
Yes, sir.
The other functional groups which are removed from the molecule because they have identified that those functional groups are not important for the biological activity. So they have identified that this functional group pH, this OH, and this NHMH3 that alone contribute for the biological activity. So they retain those 3 functional group, and those 3 functional groups should be called as pharmacophore. Okay, so I think first time I am using this word pharmacophore. Let me define what is meant by pharmacophore. It means that the atoms or group of atoms, okay, which is contributing for the biological activity of the molecule. So in this example, pH, OH, NH, Me. So these are the 3 functional groups. They contribute for the biological activity of the molecule. Hence, these three functional groups should be called as pharmacophore. And this entire structure is after simplification of the original lead molecule. Is it understandable?
Yes, sir.
Yes. Okay. So I just wanted to give some real-time example. Okay. In the previous example is just for simple understanding. So this time we take some real-time example, a compound from natural source, morphine. Right. So can you tell what is the source of morphine? So from which plant we can isolate morphine?
Opium poppy, sir.
Absolutely right. So opium poppy is the source of this morphine. So if you look at the structure of morphine, okay, so what I'm showing on this slide is structure of morphine. Can you tell whether this structure is simple or complex?
Complex, sir.
So how do you say it's a complex structure? So what makes you to
Groups, more number of links.
many functional groups are there, right? So here we have CH3 group, here N is there, here there's a OH, there's O here, another OH here. So we have how many rings there? So this is the first ring, this is the second ring, and this is the fourth ring, and this is third ring, and this is the fourth ring, right? So almost it is having 4 ring system, and it's having OH at 2 position, and it's having O at 1 position, nitrogen in 1 position, CH3 in another position, right? So all this makes this complex. Even if we look at the structure, we can say that it's a complex structure. But the complexity, what I'm talking to you is in terms of synthesis. Okay, not based on how the structure looks. Okay, based on the synthetic feasibility, we can say that this molecule is a very complex molecule to synthesis in any laboratory condition. We cannot easily synthesize this molecule in 1 or 2 steps, right? So it may go like more than 10-15 steps to finally get this final product as morphine, right? So because of the synthetic difficulty, what the medicinal chemist used to do is they go for kind of optimization, and they use a technique called as simplification, right? So if you want to simplify the structure of morphine, what we have to do first, based on what I have discussed in the previous slide?
Remove unwanted functional groups.
Okay, so remove the unwanted functional group. Okay, if you want to remove the unwanted functional group, first thing I should know which functional groups are wanted. Am I right? Then only I can say which functional groups has to be removed from this structure. So identifying which functional group is important, that is the pharmacophore. We have to identify the pharmacophore of this morphine before making this before going for the simplification process. Okay, let me erase everything. Okay. Yeah. Okay. Yes. So whatever is labeled here as the green color, those are the additional or the excess functional group. So this O here, this OH here, this double bond here, some way the medicinal chemists, they have identified that these are the functional groups which are not much important for the biological activity of morphine. Okay, so we can say this is important, this is important, this nitrogen, this CH3, these are important, but this double bond, this OH and this O, they are not much important, right? So based on this knowledge, right? So based on the knowledge about the pharmacophore, so now we can go for simplification. So let's see how they made, how they simplified the structure. Do you find any difference between the first molecule and the second molecule?
Yes, sir. They removed one functional groups.
They remove few functional groups. Okay, so can we say that levorphanol is slightly, it's slightly a simplified structure of morphine?
Yes,
Yes,
sir.
sir.
Can we expect that levorphanol will be also having similar activity like morphine?
Yes,
Yes,
sir.
sir.
So how do you say that?
Because they have removed the excess functional groups alone.
Okay, in another words, they have retained the functional groups which are important for the biological activity. Am I right?
Yes, sir.
Okay, are you able to understand? The pharmacophore is retained in levorphanol. Are you able to understand?
Yes, sir.
Yes, sir.
Yes, right. So that's why we can expect, right? So can we? So you had to think, right? So we have levorphanol. Now the question is, still this complex, this molecule is a complex molecule. We cannot that easily synthesize the molecule. So what we have to do is we should go for further simplification, right? So so this is an excess ring, and in this they remove that excess ring, and they came up with another molecule, metazokine. uh, so what's the structural difference between morphine and metazokine? So can we say metazokine as simplified form of morphine?
Yes, sir.
Still the pharmacophore features are retained in metazokine. Am I right?
Yes, sir.
Okay, this procedure is what is called as simplification. So basically why we do this simplification is to make the molecule easily synthesizable in the normal wet lab conditions. Okay. uh, okay, I just want to skip few things. This is another example for simplification. So whatever here highlighted as green color, so those are the feature which are very much important for this glypine, which is an FDA approved drug molecule. And here you can see how they remove the unwanted or excess functional group. They come up with molecule A, then further they went for simplification further, and they came up with molecule B, then molecule C. So can we say that molecule C, and we can expect the same biological activity like glypine molecule C?
Yes, sir.
So how do we say that? Because the pharmacophore feature is retained.
Retained.
Am I right? Yeah. Okay, that's good. So even further they went for simplification. Okay, they came up with another molecule D. but this is very interesting for discussion, because we can say this as oversimplification. Okay, so structure D is oversimplification. So the meaning of oversimplification is like, the complex structure is, too much simplified. That is called as oversimplification. So the point is that we should not go for oversimplification. We had ensured that we simplified the molecule to some extent, but we did not went for oversimplification, right? So we should not go for D. I'll tell why we should not go for oversimplification, right? Even though the molecule is having same pharmacofeature like glypine, we should not go for molecule like D. So this is called as, extremely oversimplification. So I just give some example, right? So what is meant by oversimplification? What is on screen now? So we have a binding site that is represented as gray color. You can see some kind of architecture within the binding site. So what are this red color and blue color? Many times, I mentioned in my previous lecture. What are they? This red color. What are they?
Active sites.
Those are the binding regions. Okay, those should be called as binding region, which is present in the target site, binding site. Okay, so here green color, red, this blue. Okay, those are the binding region. So a molecule is fitted inside this. So here there's a kind of interaction takes place between the drug molecule and the binding site. The binding region of the binding site. So here also some kind of interaction taking place. Here some kind of interaction taking place. Here some kind of interaction. So can you tell how many number of interactions are taking place in this illustration? In this specific image, how many number of? Four interactions, right? So if I number 1, 2, 3, 4. So we can say 4 type of interactions are taking place, right? So you have to keep this image on mind because I am going for next slide. Okay, so the intention of this is to show you what is meant by oversimplification. Okay. So now, can we say this modification or simplification? So initially we have structure like
Yes, sir.
this. Okay, so this is absolutely called as a simplification. So this region is important. Okay, this group is important. This group is important, and this group is important for making the interaction with the target binding site. All other groups are not much important. Okay, so we can play around this area to simplify the molecule, but we should not disturb any of this or this or this or this part of the drug-like molecule. That's the idea, right? So in that aspect, they went for simplification. They came up with this. So definitely we can say that this is a simplified version of the previous structure. So if I go one more slide, can I call this as simplification?
Yes, sir.
Yes, right? So still we don't have any issues with the molecule, right? So molecule is there, but only problem is, this area is now, there's a vacuum or nothing is filled here. This region is hydrophobic, right? That we will address later. So can we call this as simplified version?
Yes, sir.
Okay, so I just wanted to use these two images, this one and this one, to make you to understand few things, right? So here, can we rotate this bond? Will it be possible for you to rotate this bond?
No, sir.
Either in this way or this way. It is not possible. Why? Because
Cyclic structure.
of the cyclic structure. Who answered?
Kavya, sir.
Okay. So because of the cyclic, excellent. Because of the cyclic structure, we cannot rotate this. So we can say the position of this blue color atom is absolutely, static. It's going to be static. We cannot move that. It's not dynamic. Okay, at any time, at any given time, the position of that atom will be there, and at any time it can make interaction with this. That's the point, right? But if I go one step further for simplification, as is shown, now is this bond rotatable or not?
Yes, sir. It's rotatable.
Now this bond is rotatable because we have broken that ring structure, but still the molecule is having same pharmacophore feature at the same position. No issue, right? So here we have the interaction, here we have the interaction, here we have the same interaction, here also we have the same interaction. But now some possibilities arises here that because of this bond is rotatable, right? So this bond can undergo conformational change. If it undergoes conformational change, okay, so what happens, I think I don't have okay, so what may happen is, can you agree that at some point, this blue color may move away from this position because of the rotation?
Yes, sir.
So during that time, what may happen, this interaction may not take place, and that is what we call it as oversimplification. Unfortunately, I don't have the animation for that, right? So you have to have some imagination. So we are bringing this group to this position. Okay, so what may happen is that this blue color region can further move away from this. There may be a lack of interaction between these two groups. That is called as oversimplification. So these are example for simplification, right? So here, these are the Formofer feature. Absolutely, this structure is having very complex structure. It will be very difficult for you to synthesize the natural compound cocaine. So what they did is, they completely removed this ring structure. Okay, they made it very simple linear structure like this. They came up with this, the procaine. Since the Formofer feature is retained between cocaine and procaine, procaine also has very similar effect like cocaine. This is an excellent example for simplification, right? Another example, you can see that how the complex structure is converted into simple structure based on simplification concept. And a disadvantage, as I said, that we should not go for oversimplification, right? So we should know that to what extent or what limit we can simplify the structure. Beyond that, we should not go. So if we do so, your compound may start lacking the selectivity, right? So instead of binding to only one target of your interest, it can also go and bind to other targets because of the oversimplification, right? So this is what I said. Okay, so I think here I have the illustration of effect of the oversimplification, right? So the same image I explained to you in earlier slide. So here this bond is rotatable, right? So let's see how this bond rotate and how that rotation affects the selectivity of this molecule, right? So this bond is rotatable now. It can undergo conformational change. So because of the conformational change, what happened in this image? What happened?
Its activity is decreased, sir.
Okay, so this interaction is completely broken. Okay, the interaction between this two group, right? So we cannot ensure, even though the molecule is having same form feature, because of the oversimplification, this interaction is completely lost, which may lead for decrease or no activity for this compound, even though the molecule is having the same form feature. This is a very excellent example for oversimplification. Is it understandable?
Yes, sir.
Now, I am taking the same molecule. Okay, so now I am trying to fix this molecule inside some other binding site. So you should have imagination about this architecture of the binding site. Okay, so you have to imagine this. You have to keep this on your mind. So I am going for the next slide, in which, do you agree the molecule is same? Can I say that molecule which I have shown in the previous slide and this slide are same? I'm showing that molecule again.
Yes,
Just have
sir.
a look on this. The molecule is same. So do you agree that now the molecule is not binded to the same protein? It bind to the different protein. We just look at the architecture of this protein. Do you think both the proteins are same?
No, sir, they're different.
They're different. What happened is that because of oversimplification, there is a lack of selectivity. The molecule is not binding to one protein, it is binding to some other proteins. Does it make sense?
Yes, sir.
So one of the major disadvantage of going for oversimplification is that, so whenever we do oversimplification, the molecule may lack selectivity. Is it understandable?
Yes, sir.
Sir,
Yes, right. Any
Excuse me,
question
sir?
I can address? Yeah. Any question I can address?
sir, before this oversimplification step, we had another structure, right, sir, with the cyclic string.
Yes.
Even at that time, we had a vacuum.
Though I'm not addressing that part. Okay.
Oh,
Definitely there is a vacuum. Okay. We had to accept that. I'm not talking about the vacuum. I'm talking about how the simplification may lead for lack of selectivity.
okay, sir.
Are you able to understand the point?
Yes, sir.
Okay. So still, you had to find a way to solve that. I can agree. There is a vacuum.
Okay, sir.
Okay, we had to fill with some other, instead of inserting some, ring structure there, you can try to insert some group, like structure which is not so complex. You can go for bioisoster in that specific part, but it's a very complex process. Okay.
Okay, sir.
Yeah. Okay. So oversimplification, I just skip this. Okay, next is a very commonly used strategy called as rigidification. Okay, so what's the meaning of rigid? So can you tell what's the literal meaning of rigid? Rigid, flexible. So very often we use
Can't
this
be
two
modified.
terms. No, can't be modified is not rigid. What's the dictionary meaning of rigid? Vishmaya.
Very hard.
Very hard. Who answered?
Sir, Sai Ganapriya, sir.
Hmm. Anybody with the name Vishmaya? She's not this class, huh? Okay, I am not getting any response. Aparna? Aparna is this class or?
Not this class, sir.
No,
No. Okay, so let me give you example for rigid, flexible rigidification. Okay. So I think this is very simple illustration to understand what is meant by flexible. So here, say for example, you have some functional group here, and okay. So here, say you have some functional group here, you have some functional group here, and this is carbon, and this is carbon. So literally, you have here CH2 and here CH2, right? So this you might study in your 10th, 11th, 12th standard basic organic chemistry, carbon single bond is rotatable. Okay, but carbon double bond or carbon triple bond, we cannot rotate this bond. Am I right?
Yes, sir.
Okay, so because of this carbon single bond rotation, so there is possibility for many number of conformation for this type of molecules, right? So we can say this is molecule number 1, and this is the same molecule with different conformation. This is also the same molecule, and this is also the same molecule. Difference is, they have different conformation. The same molecule is present in different conformation because of the carbon single bond. So we can say whenever you have many number of carbon single bond in your drug like molecule, we can expect that the molecule will be more flexible in nature. Understandable? So here the meaning of flexible is many number of conformations are possible. Is it understandable?
Yes, sir.
But if I say rigid, which means that we cannot generate many number of conformation for that molecule. Okay, that is the meaning of rigid, flexible in terms of drugs. Okay, so see here, can we say that this bond is a rotatable bond? I hope everyone is following me. Can we say this is a rotatable bond?
Yes, sir.
Yes, right. So let me label that carbon as one second. So let me label this as carbon. We have carbon. We have literally we have carbon carbon single bond, which is a rotatable bond that is highlighted using that red color arrow. Absolutely, that bond is a rotatable bond. So do you agree that we can generate many number of conformations for molecule 1 because of the bond rotation?
Yes, sir.
Yes, right. So that's what I'm going to show you. Can we say 2 is one of the conformation of 1?
Yes, sir.
Yes, 2 is one of the conformation of the first molecule. We have carbon carbon here. So this bond undergone rotation, so which lead for compound 2. It's not compound 2, it's another conformation of compound 1. So okay, so it's a very interesting example. So we have receptor here. So the receptor architecture is like this. So the receptor binding site is having some binding region here. This yellow color region is the binding region which is present in the binding site, and this OH group and this COO minus. Okay, so these are these are some other binding groups which are present in the receptor. So the ultimate aim is that we have to try to fix the molecule 1 or its another conformation, that is 2, inside the binding site. So which one we can fit, or can we fit both the molecules? So one we can. Sorry, somebody is trying to answer.
We can fit one, sir.
Okay, we can easily fit one, right? So I do agree. What about 2? Can we fix or not? No. Okay. So as you said, in this we have fitted molecule 1. See here, that's a kind of interaction. I have another, receptor 2. So the architecture of receptor 1 and the architecture of receptor 2, they're completely different. So can we fit molecule 1 in this binding site? No. Can we fit molecule 2 in this binding site?
Yes, sir.
Yes, right. So this region of this will interact with the yellow color region. So this OH group, the oxygen, can make hydrogen bonding interaction with this group, and this positive charge NH2Me can interact with the negative charge COO minus. That is illustrated here. Okay, so we can fit, right? So what is the selectivity of this molecule now? Is this molecule selective or not?
Yes, sir.
Yes. First you have to understand my question. Don't be very fast. My question is this molecule selective or not? At least at this point, you should know what's the meaning of selectivity, because I gave a lot of examples in previous slides. Selectivity is the molecule binding to
No, sir.
Okay, no. Okay, so I hope that everyone should agree that no, this molecule is not selective, because the same molecule is binding to receptor 1 and it is binding to receptor 2. We cannot say that the molecule is selective. Say for example, we need selectivity for this molecule. Okay, so assume that the molecule has to bind either to receptor 1 or to receptor 2. By any chance, it should not go and bind to receptor 1 and receptor 2. We are interested to bind this molecule either to receptor 1 or receptor 2. So what we can do is we can go for a method called as rigidification. Okay. So just see here what we do in rigidification. So which of the two molecule is flexible? So can we say that the first molecule is flexible and second molecule is rigid?
Yes, sir.
So how do you say first molecule is flexible? Because we can generate many number of conformation of the first molecule, but the same thing is not possible for second molecule because of the cyclic structure here. Do you agree?
Yes, sir.
Yes, right. So now which of this molecule 1 2 is selective?
Second one.
Second molecule is now selective. So how we achieved the selectivity? What is the method? What is the strategy used for selectivity?
Rigidification.
Okay, that's all. So that is what is called as rigidification, right? So hope everyone understand. Absolutely, that is what is called as rigidification. So here I have some more examples. All story is the same, right? So we convert the flexible molecule into a rigid molecule. That's what's happening in all the examples. Okay. I just want to skip. So this is the example for the methods used in rigidification, right? So you can insert a double bond to make the molecule highly rigid, or we can insert a ring system like this to make the molecule rigid. We can also go for a triple bond system or CONH, right? So this is very interesting. So can you tell how this is rigid? Still we have single bond, no?
Partial double bond, sir.
Who answered?
Vasily, sir.
Okay, absolutely right. So because of the partial double bond of this amide, okay, this bond is called as amide, CONH group, right? So this is the primary bond you can find in the proteins formation of if you join many number of amino acids through this amide bond, right? So this is rigid even though it looks like single bond, it is not single bond. So this has characteristics of partial double bond. What she said is absolutely right. So these are the different methods, simple methods we can organic chemistry methods we can use for making the molecule rigid. Is it understandable?
Yes, sir.
Yes. Okay. Okay. uh, all these are example, right? So you just go through, you can easily understand. So so this is the original molecule. So second molecule, can you tell how they achieved the rigidification? How they converted the flexible molecule into rigid molecule? What's the approach used here? They inserted a triple bond, right? So here how they achieved that? It's still more here. What's the method they have used? They have inserted the amide bond. Okay, so again this amide bond is highly rigid. Also, the flexibility which is present here also highly restricted by inserting the ring system. Okay, so multiple ways we can do that. That's the point. So another example, right? So these are real-time example you may go through, right? So when we go for this type of rigidification, we cannot guarantee that the activity will be retained. The example in which E isomer and Z isomer of this cumbertastatin, it's a kind of statin molecule which is used for controlling cholesterol. So what they have identified is the E isomer is having very less activity comparing to Z isomer. Okay, so which means that whenever we play around this concept of rigidification, we cannot guarantee that the biological activity will be improvised. Okay, so those and all we had to address later. Okay. Anyway, I am not touching that now. I skipped many slides, not much important. Okay. these are just graphical abstract of what I have discussed. I think that's all for lead optimization. Any question I can address?
Sir, how do we find the pharmacophore of a compound, sir?
Okay, that's a very good question. Let me answer. Okay. So, is OneNote visible for you?
Yes, sir.
Yes. Okay. So, how to identify the pharmacophore? So, I just want to take the structure of morphine here again. Let me take the structure of morphine. Uh, just a few seconds. Okay. Let me draw the structure. Or I think I have some other drug structure. Let me take that picture. Okay, desktop, and aspirin. Okay, we have two structure. We'll take that. Is the structure visible for you?
Yes, sir.
Okay, so okay, say for example, this is a drug molecule. We'll take this as an example, right? So it will be simple for me to make it understand. Is the structure clearly visible?
Yes, sir.
Okay, so how to identify the pharmacophore is the question. So it's not a difficult thing, right? So it's a primary, experiment done by medicinal chemist with the help of the pharmacologist or microbiologist, sorry, molecular biologist. So pharmacophore is basically the atom or group of atom that is responsible for producing biological response. Okay. So how to identify the pharmacophore element? So first, what we have to do is like. Okay, first what we have to do is first we had to identify what are the major functional groups which are present in this molecule. So for example, we have COOH group, we have CH3O group is here, CH3 is here, Cl is there here, O is here, N is here, and some ring systems are there, right? So what we have to do is we have to start with any one of the functional group at a time. So first, what the medicinal chemist does is that, say for example, they replace this. Let us assume that this group is removed, and this group is replaced by hydrogen. Okay. So before the replacement, this molecule is an anti-cancer drug molecule. Okay, the activity of this molecule is anti-cancer activity. So now what they did is they replaced this Cl with H. Now again, for this analog, they evaluate what's the biological activity, whether the same biological activity is retained or not. Okay, so if there is no change in the biological activity, what does it mean? What we can infer from this experiment? I changed the Cl to H, but there is no change in the biological activity. What does it mean?
It is not a pharmacophore, sir.
It is not a pharmacophore. It is not contributing for the biological activity. Okay. So say for example, while replacing the CH3O to H, you can replace with some other group also. Just I am taking simple example to make it understand, right? CH3O, right? So if replacing the CH3O with H, say there's a 50% decrease in the biological activity. What does it mean?
So it is important for biological activity.
It is important for biological activity, right? So this is the way how we can discover the pharmacophore. Is it understandable?
Yes, sir.
Yes, this is the way how we can identify the pharmacophore elements. So any other, I hope I have answered. If any more doubt, I can answer.
So you told me so they'll check the activity after replacing various components.
Can you say your question again?
So now only just told me so they'll just replace some things and they'll check the activity of it.
Mm-hmm. Yeah.
So will they use some computer software or will they go for wet lab techniques?
Okay. That is also an excellent question. In both the ways we can do that. We can do this in wet lab assays. Also, we have some software. By using that, we can find the pharmacophore. What I mean here is the pharmacophore can be identified using wet lab. That's what I explained. Replacing this. Also, we have some computational methods for identifying that. There are a lot of softwares which are available. We can make use of that. What I mean here is using both wet lab as well as computers. We can to some extent identify which functional groups important for the biological activity.
Okay, sir.
Yeah. I think any more question? I think next topic I'd like to start from next lecture class. Any question I can discuss now?
Sir, this is Gayatri, sir.
Yes.
Sir, I have a question. Does lead optimization influence the half-life, biological half-life of a drug?
Is it your question or statement?
I'm asking you, sir, does lead optimization influence the biological half-life of a drug?
Half life, will it affect the half life or
Yes.
will it improvise the half life, right?
Yes. Sir.
Lead optimization, if okay, so say for example there may be some drug like molecule you may work with, right? So say for example if you take many polyphenol.
Okay, sir.
Okay, so normally they used to have good activity,
Okay.
but very unfortunately the half life of those polyphenols, so many polyphenols we can name, right? Say for example if you take resveratrol, quercetin, so these are polyphenols, flavonoids. So if you check the half life, t half of those molecules, normally those molecules used to have very less or very poor t half life of the molecules are very less.
Mm.
So in that case you can go for lead optimization and you can improvise the half life. That is possible. Okay, there are many success stories, many papers you can find. If you even do Google search, you can see what is the technique used by the medicinal chemist for improvising the half life of drug molecule. Okay, the reason for decrease in half life is related to its metabolism, rapid metabolism. Okay, so the medicinal chemist will work on improvising improvising the stability of the molecule to some extent, and that will eventually lead for improvising in the half life.
Okay, sir.
Okay, so we can do lead optimization to improvise the half life of the drug like molecule.
Okay, sir.
Yes.
So I have
Any more
one more
question?
question.
Yes, go ahead.
Does these modifications, the simplification, rigidification, other modifications, cause any side effects?
It may cause or it may not cause.
Okay, sir.
But whenever we make the replacement, it's not trial and error method. It's a very scientific procedure.
Okay.
As a medicinal chemist, we know that which functional groups will cause toxicity.
Yes, sir.
Okay, so while making the modification, we won't prefer those functional groups.
Okay.
So whichever functional groups are already reported as a very safe, which won't much affect the biological system. So those are the functional groups which are chosen.
Okay, sir.
Okay, so it's a kind of very controlled, very scientific, way of modification. It's not random.
Okay, sir.
Yes. Any
Thank
more
you.
question I can answer? Yeah. I think we have 5 more minutes. We can make use of discussion. Any more question I can address? Any question related to today's lecture or previous lecture or any general questions? Any more questions?
No, sir.
Okay, so if no more question, I think we shall wind up the session. Okay. Okay, thank you. We will see in the next session.
Thank you,
Thank
sir.
you, sir.
Yeah, thank you.